Hi, my name is Jennifer Flynn, and I wrote my paper over the enzyme Lysozyme. Now, everything a person does happens through a mechanism that happens in the body prior, and these mechanisms are made possible by proteins. Proteins are molecules that are made up of amino acids, and they carry out many functions throughout the body, such as generating movement, storing molecules, transporting molecules, even gene regulation. And as an example, I brought a necklace, and I'm going to use this to explain the structure of a protein. So the amino acid is, consider that one pearl, and in the amino acid, it's a carbon attached to a carboxyl group, an amino group, an R group, which is what makes the identity for each specific amino acid, because the R group changes between each one. It's different for each one. And a hydrogen. The hydrogen will be used later in hydrogen bonding, which will be for the secondary structure. But for now, we're on the primary structure. So the primary structure is going to be for just an amino acid. It's going to bind the carbon of the carboxyl group to the nitrogen of the amino group. Now, whenever you move to the secondary structure, you're still going to need these R groups and the hydrogens, and that's what you're going to be able to use, the hydrogens at least, to form the secondary unit of structure, which is an alpha or a beta helix. And an alpha helix can be described as a winding staircase, and a beta helix, the best thing I could think to describe that would be like folding a towel, how it just kind of lays on itself and folds over and over. And or maybe waiting in a long line at like a park or something, and for a ride, and it goes back and forth, and it kind of just trails back and forth side by side, and that is going to be the helices, and that is going to be for the secondary structure. Now, going on to the tertiary structure, in the secondary structure, those occur through hydrogen bondings, which is again where the necklace plays in. So if one amino acid is the pearl, then you could think of the string between the pearls that hold each pearl together as the hydrogen bond for each molecule, for each amino acid or protein. And next would be the tertiary structure. So the tertiary structure is kind of like the whole structure of the molecule. It includes the primary structure, the amino acid backbone. It includes the secondary structure of the helices that create the shape of the backbone, and then but it's all kind of folded into one, and it uses several different kinds of bonds, disulfide bonds and hydrogen bonds. Again, it uses peptide bonds and also R group bonding, which is where I said that each R group is different and unique to each protein. So if you look at the necklace and you take it and you knot it up and you kind of just make it messy, that can be thought of as the tertiary structure, because you've got all the amino acids, you've got all the bonding, and you've got you've got the secondary bonding where the helices, how it turns, but you can think about the knots where it turns and it hits itself and it kind of overlaps and all that as the tertiary structure, where the disulfide bonds and the R group bonds are present. And then a quaternary structure is if you take two necklaces, so two proteins, and you kind of just fold them together and you knot them up, and just like the knots were in the first structure for the tertiary bonds, it's going to be the same for the quaternary bonds. Now, back to move on to my protein, I had enzyme. I had lysozyme, which is an enzyme, and its primary structure is it doesn't go all the way to a quaternary bond, because it only bonds, it is a bond to another molecule, and it is it's a really important molecule for the immune system, because it fights off a lot of bacteria, and it fights off it helps with hygiene basically, and antimicrobial benefits. So it's in your saliva, it's in your tears, it's in your sweat, and in your any type of mucosal secretion, like nasal secretions and everything. It's going to be in there. That's actually how it was discovered. Alexander Fleming, the same scientist who discovered penicillin back in the 1920s, he discovered lysozyme just prior to discovering the penicillin antibacterial qualities whenever he learned about it with fungi. And he learned that whenever drops from his nose accidentally fell into an agar plate from a cold, the agar plate did not grow protein, which is later what he found in penicillium, the penicillium fungi. Now, the 3 levels of structure for the for the lysozyme enzyme are not, just going to go to tertiary. I think I said that already. But that's what's going to help it function properly, because it's not binding to another. It doesn't rely on another protein to do its function. It can do it by itself, and it can finish its function by itself. And I have some pictures of the shape that it takes, and I'm going to bring them up real quick to display them. There is a ribbon shape, a globular shape, and a space filling model, which is the globular shape. Oh, I'm sorry, the stick model is the, you can't see that very well. So this one right here is the stick model. This is the ribbon, and this is the globular. The stick model displays the bonds between each specific, like the hydrogen, the disulfide, the R group. The ribbon model displays the helices, and the space filling model just kind of shows each and every element like as a little balloon or a little ball, and it just fills the entire space and makes that globular protein, which earlier is what I said was the quaternary structure. This would be the globular protein of the pearl protein that I displayed. And then, let's see. So in the body, there is a gene, and it's called the alpha-lactalbumin gene, and it is the gene that codes for the creation of the lysozyme enzyme in the body, and that code comes from the chromosome 12. And so if there's a defect in chromosome 12, then you won't be able to make the alpha-lactalbumin, which would prevent you from making the lysozyme, which could potentially cause a lot of different types of autoimmune deficiencies and amyloidosis and all kinds of different ailments and sicknesses, and it would be really hard to live a healthy life without it. So in conclusion, enzymes are important, but lysozyme specifically has a lot of important roles to play in health, in healthcare, and in medical use, because I know they're doing a lot of research with it right now to try and pinpoint the work that it does on an enzyme. And so this video has gone way too long, so I'm really sorry about that, but I was able to get everything in there, so thank you.